So we'll take a look at some of the ways that animals are divided into major groups with some additional terms besides phylum categorizations. So the first term here is metazoa. So all animals are considered metazoans. And the first division into different groups is parazoa and eumetazoa. And this difference is established based on the presence of what are called true tissues. So eumetazoans have true tissues, parazoans do not have true tissues. So what is a true tissue? A true tissue is a specialized layer of cells that have a coordinated function. So for example, skin tissue or stomach tissue, they have multiple layers of cells piled on top of each other, and they work together to make an organ function usually. And so in sponges, the earliest type of animal, they do not have specialized tissues or organs. The phylum name for sponges is Porifera. Because this is the only phylum within Parazoa, these terms are oftentimes used interchangeably. So because they don't have true tissues in the parazoans, an interesting thing about sponges is that you can actually take apart the cells in the tissue and they will reaggregate back together and still be functional. So you can actually take sponge, blend it up and separate sponge cells, and then they will come back together. The other outcome of not having these specialized tissues and organs is that they have irregular body forms. So they lack any sort of symmetry or regularity in their shape. So sponges, two sponges that are the same species can look different from each other. So this is a sponge body, and we see that there's not really any way that you could divide this body. It's just kind of these vase-like shapes in random orientations. And again, eumetazoans have true tissues, at least one type of tissue, and oftentimes organs, and eumetazoans include all other animals besides sponges. So we have our metazoans, and we divide that into parazoa. Actually, let me do eumetazoa over here. We divide that into eumetazoa and parazoa. And remember that difference is based on eumetazoans having true tissues. And then the eumetazoans are divided based on symmetry. You have your radially symmetrical organisms, so organisms with radial symmetry called radiata. And then you have your organisms with bilateral symmetry called bilateria. Okay, so this is a separation based on symmetry. They're either radially symmetrical or bilaterally symmetrical. That's where their names come from. So a radially symmetric organism, members of radiata, are able to be divided in any orientation. So if this is a jellyfish, for example, I'm looking at the top of the jellyfish here, and I could cut it in any place like where these line hash marks are, and I would get half of the organism. And so if you think about an animal like us, this is not true. You cannot cut in multiple places and get symmetrical halves in a human. Instead, you would have to only cut down this top center. That would be the only way that they would be two symmetrical sides. And that's because we are bilaterally symmetric. So here's another example of a bilaterally symmetric organism, in this case a crab. And notice that the crab could only be cut down in this angle to get two equal halves. If you cut it here, it would not be an equal half. If you cut it here, it would not be an equal half. If you cut it here, it would not be an equal half. So this is a bilateral organism. Most animals are bilaterally symmetrical, okay? The only organisms that are not are the jellyfish and their relatives. Let's see here. Oh yes, so let me show you one that people get confused about. So this is my really, really lovely drawing of an octopus. And so from my kind of ugly drawing, you can see that a jellyfish and an octopus in many of our minds have a somewhat similar shape. And so I want to remind you that, in fact, an octopus, however, has 2 eyes and a beak, a mouth type structure, and that, in fact, for that reason, I should add there's also a brain in there. For that reason, you can actually only cut an octopus one direction in half. It's not like a jellyfish. Jellyfish have no eyes, no nose. They don't have that same kind of body orientation that we have. So there's another word here I want to mention, cephalization. A jellyfish does not have a head. That's cephalization. Cephalization means having a head. An octopus does. And so cephalization actually goes along with being bilaterally symmetrical. Organisms that are cephalized or have heads are bilateral. Radial animals do not have heads. And so instead they actually have a mouth side, which is actually on the bottom usually, and a top side, and are usually circular or tubular in shape. So in this case, we have this sort of circular jellyfish. Another example that you may be familiar with is a coral or a, blanking on the name, anemone, sea anemone, which have more tube-like shapes. So the symmetry of organisms correlates with the number of cell layers in the embryo. So when we say cell layers, we're talking about during embryonic development. So to just overview embryonic development a little bit, you have a zygote, which is a fertilized egg, and that begins to go through a lot of mitosis. So the cells begin to divide rapidly. This is an embryo at the 8-cell stage. And then as more and more cells form, the cells take on this form. It's called a hollow ball, or the blastula. So there's no cells on the inside. All of the cells have migrated to the outside. And this will eventually fill in through a process called gastrulation. So cells will continue to divide, and then they will pass through the gastrulation groove right here, and then they will start to fill up the inside of the hollow ball. And so this is where the difference arises. In bilaterally symmetrical organisms, the cells will fill in and form 3 layers. So this is a bilaterally symmetrical organism, also called a triploblast, or being triploblastic, triplo for 3. So it's actually forming 3 cell layers. There's that outer layer that was the blastula. That's called the ectoderm. There's this endoderm, which is this inner layer here, and then these red cells are populating what will be the mesoderm, the middle layer. So the endoderm is the inner layer, the outer layer is the ectoderm, and then bilateral animals have that middle third layer called the mesoderm. And that mesoderm will actually go on to form muscles and complex organs. And so bilateral symmetry is also associated with having true muscles and more complex organs than you see in radially symmetrical organisms. Because radiata are just diploblastic, they're only going to have 2 German layers. So again, they're only going to have these 2. Okay, so let's again draw our animal categories. We have our metazoans, divided into Eumetazoa and Parazoa. And then we have our Eumetazoans divided into Radiata and Bilateria. And then our Bilaterians are going to have a major division into protostomes and deuterostomes. And this division into protostomes and deuterostomes also has to do with a feature of embryonic development. This is a different feature of embryonic development. So while the cells are dividing in the early embryo, the cells stack on top of each other. And so how they stack on top of each other is referred to as either spiral cleavage or radial cleavage. So in spiral cleavage, the new cells are stacking in between the old cells. So if you look, these are the old cells, and the new cell is stacking in between them. Whereas in radial cleavage, the new cell stacks right on top. So see, this is the old cell. A new cell has stacked right on top of it. And so the outcome of this, and this is actually the more interesting point, somehow relates to determination. So let me explain a little bit before I get into the difference. Cell fate determination is when a cell moves from being a stem cell that has the ability to become anything. So pluripotent means multiple potentials. It has the potential to become anything. So stem cells in the early embryo are pluripotent. They can become any type of cell in the body. And through determination, they will get turned into regular cells that now have their fates determined. That means they can only become certain parts of the body. So in other words, they're no longer stem cells. They can only become, for example, a skin cell or an eye cell, depending on the level of determination. This process of determination starts to happen very early in the embryo. So, early patterning, like you can only be the right side of the body or you can only be the left side of the body. Then it starts to get more detailed, like you can only be the skin or you can only be the eye. But depending on the point in which you are at in the embryo, the fate has been more or less determined. Okay. And so also depending on the type of organism you are, the fate has been more or less determined at different points in the embryo. And so in protostomes, they have what's called determinate cleavage. And so determinate cleavage means that at the very, very earliest stage of the embryo, just at the 4-cell or 8-cell stage, the embryonic cells are already determined. They're already limited in what they can become. So the consequence of this is that if you have a 4-cell embryo and you take away one of those cells, so when it crosses out, I take away one of those cells and only leave these 3 cells. These 3 cells are now missing a critical cell because this cell already had a job assigned to it in the embryo. So this 3-cell embryo will die. Okay, so the fate determination in protostomes is so early in the embryo that if you take away one of the cells, then they don't have a critical component that they need, and this will not be able to develop into an embryo. Deuterostomes, on the other hand, have indeterminate cleavage. In that early embryo, the stem cells are still pluripotent. That means they can still become anything. So if you do the same experiment where you take away one of those cells and you have just this 3-cell embryo, the 3-cell embryo doesn't matter or doesn't care because it still has the ability to have these cells become anything. So this will become a normal embryo. And so this is actually the basis of identical twins in humans. So what happens is an early embryo splits in half. So this would accidentally split into pieces. They don't even have to be even, right? And this was one embryo. It accidentally splits in half and becomes 2 embryos, and this would be 2 identical twins in humans. So humans must be deuterostomes. Identical twinning can only occur in deuterostomes. In protostomes, if the embryo breaks in pieces, then the embryos all die. They're not viable. Again, because the fate of the cells is determined very, very early, whereas in deuterostomes, the fate will be determined later on. In fact, the fate won't be determined until we start filling in the embryo. So yes, the fate will eventually be determined in deuterostomes too. I want to be clear there, but it's later on than in protostomes. The other difference is that a part of the embryo called the blastopore, that opening where the embryo is folding in and filling in, becomes different things. In the protostome, that opening called the blastopore becomes the mouth, and in deuterostomes, that blastopore opening becomes the anus. So another difference in that embryonic development. Okay, so another major division. So let me sketch in my divisions here. Oops. Parazoa, Eumetazoa. Then our eumetazoans are split into radiata and bilateria. And then our bilateria is split into deuterostomes and protostomes. And then the last division we're going to talk about now is protostomes divided into ecdysozoa and lophotrochozoa. So this difference is one of the differences that has been really recently distinguished because this was established using molecular evidence. And so the physical traits were then associated with that. And so the physical traits are a little bit weird, and you'll see what I mean here. So ecdysozoa is named for the trait of molting. That process is technically called ecdysis. So molting is when an organism molts or sheds some kind of outer coating. So in some organisms, this is what's called a cuticle. The one people are more familiar with is an exoskeleton. So insects, for example, have an exoskeleton that they shed. They're members of ecdysozoa. Lophotrochozoans have one of two possible traits. Members of Lophotrochozoa either have a lophophore, which is a type of feeding tentacle, or they have a specific type of larva, trochophore larva. And so you can see what I mean about how this group seems sort of counterintuitive because they have either this or that. But remember that molecular evidence supports that they are one lineage, even though the traits that hold them together is a little bit fuzzy. So the traits for the Lophotrochozoans is either these feeding tentacles, or this special type of larva called a trochophore larva. And here we see a dragonfly molting its exoskeleton. So it's a member of ecdysozoa.